Hello students, I am Dr. Vinod Shinde. In the previous lecture, we have discussed about the sterilization method by physical agent, that is cold sterilization method, radiation specifically. And now we will discuss about the another sterilization method, that is filtration, used extensively to sterilize liquid or gases. The filtration method is used for sterilization where human or animal serum is in the culture medium is essentially coagulated by heat. Because of this, we have to use a filtration method that is having no heat or temperature. Uh, so enzymes, some toxins and antibiotics are also destroyed by heat. So these equipments or these chemicals are to be sterilized using filtration method. Such solutions are best sterilized by filtration. If filters remove bacteria naturally, they will also remove larger microorganisms or larger organisms. A number of bacteriological and serological filters are available for this purpose, and some of these are the porcelain filters, diatomous filters, filtered glass filters, asbestos filter, ultra filter, and these are the filters used. The porcelain filter are filters with Camberland filters, also called as Camberland filters, and composed of hydrous aluminum silicate or kaolin, and baked at a temperature as high as possible without sintering the clay. The filters are prepared in various degrees of porosity from 0.65 micron to 15 micron. They are cleaned by heating to burn the organic matter in the pores. The diatomous filters are prepared of diatomous earth, which is fine, usually white, siliceous powder composed of chiefly or wholly the remains of diatoms and called as clisagor. It is prepared by mixing diatomous earth, asbestos, and organic matter such as plaster of Paris in different proportions depending upon the size of pores required. This mixture is kept at high pressure and baked at 1000 to 2000 degrees Celsius to get binding. This is called Brickford or Mendler filters. These filters are cleaned by washing. Filtered glass filters are filters formed by heating powder glass in the disc form to a temperature just below the melting point. This causes the particle to adhere to each other without damaging the porosity of the disc. The disc is then carefully fused with the glass funnel and cleaned by the treatment with sulfuric acid containing sodium nitrate. The strong acid quickly oxidizes and dissolves the organic matter. The acid is then removed by thorough washing. Second is the asbestos filter. These are composed of matted asbestos fibers in the disc form and are called sieve filters. The disc is clamped tightly between two smooth metal rims and by a screw clamp. Bacteria and other suspended particles are absorbed by this filter disc and these are discarded after use. The ultra filters are next. These are composed of cellulose esters and called polypore or millipore filters. These filters are discarded after use. These are called membrane or molecular filters. These are prepared as circular membrane of about 150 micron thickness and contain millions of microscopic pores of very uniform diameter. Filters of this type can be produced with known porosity ranging from 0.01 to 10 micrometers. These filters are used extensively in laboratories and industries. In recent years, a new type, that is high efficiency particulate air filters, are developed, which are made it possible to deliver clean air in the enclosed rooms such as our cubicles. This type of air filtration, together with the system of laminar airflow, is used extensively to produce dust and bacteria free air. Now, the last point in this chapter that is microbial death concept we'll discuss. The term death is defined to in irreversible loss of ability to reproduce. Viable microorganisms are capable of multiplying. Dead microorganisms do not multiply or grow. The failure of microorganism to grow when inoculated in an appropriate medium indicate that the organism is no longer able to reproduce, and inability to reproduce is considered considerable criteria of death. When a drop of suspension of bacteria into little in a bottle of hot acid or an incinerator, the bacteria may be killed so fast that it is not possible to measure the death rate. Exponential death can be understood easily in terms of simple model. Imagine, for example, each cell is a target and large number of chemicals and physical agent as a drop or molecule are sprayed on them in a random manner. The probability of hitting the target is proportional to the number of targets. The number of bacteria in this case, as there are many target bacteria killing by the chemical or physical agent that will kill majority of microorganisms, but as time goes, the number of bacteria in the suspension decreases, leading to the point where chemical and physical agents will find hard to kill remaining one or two bacteria. The probability of killing bacteria by chemical or physical agent is proportional to the concentration of the agent, that is chemical or physical agent. If bacteria are killed by physical agent, the bacteria are killed very fast as the intensity of X-ray or UV light increases. In the case of chemical agent, the cells will be killed more rapidly as the concentration of chemical increases. If the population of cell is higher, that is contamination is higher, the time required for killing by any agent, that is chemical or physical agent, will be more if the concentration at any concentration. Therefore, we have to treat a contaminated site for longer period of time. The factors influencing the death concept or death of microorganism is number of microorganism. If the controlling agent or physical agent or chemical agent used in low concentration and number of bacteria are higher, then the organism there is killing of microorganism but does not is not done efficiently, and if the agent is are not having specific concentration, then the microorganisms are not get killed efficiently. Then the next is the kind of microorganism. Microorganisms in the vegetative cell form are killed efficiently than the spore forming cells. These cells are resistant to chemical or physical agent and also the susceptibility of organism towards the antimicrobial agent to be used. The thickness of cell wall and encapsulation of cell also increases the resistance of cell towards a chemical or a physical agent. And then the third point is the environment that in which you are using the chemical or physical agent. The efficiency of the physical or chemical agent depends on the environmental condition, that is effect of radiation will be lowered if the room is filled with the dust particles. Chemical composition of the medium containing microorganisms will be affected if the chemical agents, as it will react with the chemical agents to form products which are less antimicrobial. Chemical composition of the reacting medium also changes the chemical efficiency of the chemical to be used for killing purpose, and it will change the product efficiency. Organic matter present in the environment will influence the activity of the antimicrobial agent, and organic matter will react with antimicrobial agent, precipitating the antimicrobial agent, leading to lowering of the effectiveness of the antimicrobial agent by lowering the concentration of antimicrobial agent. So, students, this is the chapter that is physical and chemical agents used to control the microorganisms in a medium or in an environment. So, thank you very much.